All right, here, this is a little different. Uh, there's one group of organisms we're looking at. We use as an example the chicken embryo, but this is an evolutionary lineage called the amniotes. They have a slightly different developmental pattern because all the amniotes, which are reptiles and mammals, birds are in there with the reptiles, they have that huge amount of yolk at the bottom we looked at during cleavage. So the cells don't even divide completely. You get this big mass of yolk at the bottom. The embryo just divides at the top. So they have the incomplete cleavage. And this next stage after the morula also looks a bit different. Now, what this thing is, they actually use a different name. Note up here, this is called a blastocyst, not a blastula. Functionally, they're very similar, but there are some differences in the detail and when they sort of worked about. So it's got a slightly different name. So blastocyst will distinguish this stage in the amniotes as opposed to this stage in the sea urchins or the frog embryos. Now, a couple parts to look at. This is still more or less, a hollow ball. You've got the hollow space here, still a blastocele. But then you've got a layer of cells on the outside here. They call that the trophoblast specifically. We didn't have a trophoblast in the echinoderm or frog embryos. And then, use a different color, you've got this red shaded mass of cells here. That's called the embryoblast, or sometimes it's called the inner cell mass. The reason for the distinction, in the frog embryo and the echinoderm embryo, all of those cells will be part of the organism. They'll be its spines, its legs, its circulatory system, whatever it has for a circulatory system. All part of the embryo. In the amniotes, not so much. Just this embryoblast. If you look at these cells and you follow them, they will be the embryo's pieces: its arms, its legs, its guts, its head. All the rest of this out here, the trophoblast, well, genetically, they're embryonic cells. They are not going to be part of the embryo, none of its pieces, not its arms, its legs, its guts. They're going to end up producing some things that are, we'll just collectively call them extra embryonic membranes. These are some support structures that are only found in the amniotes. Things like the amnion, which encases it in fluid, a yolk sac that holds that relatively inert yolk, a chorion, an allantois for waste material. So these are genetically the embryo's cells, but they're not going to be part of the embryo. So that's why we have a bit of a distinction in the name, and we got a few more parts. All right. Now, this is just a sequence of diagrams showing what's a human embryo. A chicken would look about the same. But you'll notice over here on your left, that's the morula, solid ball of cells still encased by the fertilization membrane. And I mentioned in the lecture that fertilization membrane allows you to judge the size. The thing doesn't grow. This also prevents implantation in like the uterine tubes in placental mammals. This embryo will not implant in the uterus until it sort of hatches out of the fertilization membrane. What you're seeing here, this is an early blastocyst. Right here. That's the inner cell mass. That'll be the trophoblast. Now, what we talked about at this stage, in a human, and all the other blastulas of the other organisms we looked at, these cells that form this inner cell mass here, or embryoblast, these are what they're referred to as the embryonic stem cells, or indeterminate cells, or totipotent cells. Those terms kind of somewhat interchangeable. These are cells, there's a little controversy. There's some ethical issues with them, particularly if it's human. But the importance of these cells, and the reason they might want to use them for research, these cells genetically have not lost access to any of the genome. That is, any of these cells can produce any tissue. In fact, this is where you can get, identical twins. Humans have two types of twins. You got fraternal, which is just two eggs fertilized by two sperm. They're just siblings, like any others, just happen to be born at the same time. Identical twins occur if you damage or separate clumps of this inner cell mass. One half would develop into a complete individual. The other half would develop into a complete individual. And they're genetically identical because they come from the same sperm and eggs. So the idea with these indeterminate cells, term I like to use, their developmental fate has not been determined. They could produce anything. So in theory, what they want to work on maybe down the road is, you got some cells dead in your brain or your heart. Get some of these embryonic cells, somehow convince them to develop into, cardiocytes or neurons or whatnot. But that's the idea. Now, in humans, once you get to the late blastocyst stage, what they're showing here, the third one in the series, the cells lose that characteristic. They're no longer indeterminate cells. Their developmental fate starts to kind of become locked in. Basically, they lose access to the genome. Eventually when cells specialize, like a skeletal muscle cell, it has a complete genome, but it doesn't look like any other cells. It doesn't look like an epithelial cell or anything like that. Why not? Well, because think of it, the genes that produce things like teeth and fingernails and hair, do you really need those in a skeletal muscle? It'd be kind of uncomfortable to have teeth and hair and stuff in your skeletal muscles. So basically, part of the process of specialization is the cells lose access permanently to some of the genome, basically the parts they don't need. So what we've got at this point, unlike the other embryos, the frog and the sea urchin, that's pretty much what you are at this point. You got this layer of cells they've shaded in blue, layer of cells they've shaded in yellow. The reason they're using the color, at this point, the cells are starting to form the embryonic germ layers. Now, I went into a little bit more detail, but the embryonic germ layers, these are groups of cells that are beginning to lose access to part of the genome, and they can no longer develop into a whole embryo. There are 3 embryonic germ layers in amniotes in humans. I'm just going to real quick give you their name and their function. Now, at this stage, you've only got potentially you got a couple of germ cells that are formed, and we'll get a third one in the next stage, gastrulation. You'll have all 3. But the 3 germ layers are the endoderm, which is shaded typically in yellow on these diagrams. Endoderm will produce the digestive systems and associated structures, liver, intestines, pancreas, gallbladder. And also, if you have, like in humans, a respiratory tract, you have lungs, that also develops from the endoderm. This blue area, this is representing ectoderm. Ectoderm will produce your integument, your skin, sort of the outer covering, as well in the humans and chordates, it produces the nervous system. So if you grew those ectodermal cells, if you could culture them or grow them to completion, all you get is a kind of a bag of skin with brains and spinal cord and nerves in it. That's about it. The third embryonic germ layer, which does not appear, I'll look at this last one real quick, right here. This is the third embryonic germ layer, these little red cells. Those are cells that migrate in from the top. They actually started up here, what we'll call a presumptive ectoderm. It's not quite ectoderm yet. They migrate from the surface, top surface, down into the space between the presumptive endoderm and ectoderm here. Those are going to be the mesodermal cells. That's the last of the embryonic germ layers. That produces basically everything between the guts and the skin. It produces the muscle, skeletal muscle, the bones, the blood vessels, the heart, the kidneys. So kind of everything in the middle, mesoderm. Now, the next stage in development, we're at the blastula or blastocyst in the case of an amniote here. The next stage is called a gastrula. The process is gastrulation. We'll look at gastrulation in all 3 real quick, and I'll come back to the amniotes last because they're a little trickier. All right. Now, what you're seeing here, this is an echinoderm, and remember, it was a hollow ball of cells, the blastula. The next stage is gastrulation, and what you'll notice is down here at the bottom, these cells, they kind of grow differentially, wider at the top, smaller at the bottom, so they kind of grow into this little wedge, and that pushes the floor up a little bit. Then you get some cells that grow these long filamentous, they're called filopodia. They're just extensions of the membrane and cytoplasm, long little threads. They reach up to the opposite end of the embryo here, attach, and then they contract, and notice they pull this whole tube. So it's almost like, the analogy is a balloon and you punch your fist into it. That's what the gastrula looks like in an echinoderm. You get this tube just pushing up, and as it pushes up, again, you see that color change. So that fate mapping. This tube in yellow here, that's the archentron. One of these openings will be the mouth. It'll form an opening down here, which will be the anus. So that's going to be its digestive tract. The archentron is just primitive digestive system. And these cells are going to form intestines and so forth. They're endodermal cells. This opening is called the blastopore, be the mouth. This is still blastocele out here, this hollow space, but it gets smaller and kind of less important as we move on. Now, some of these cells, you notice these red ones here, they have migrated into the space in between. Those red cells are going to form the third germ layer in the echinoderm, which is the mesoderm. And so what happens in gastrulation, you basically form the 3 embryonic germ layers, so they'll have some cells from the endoderm, mesoderm, and ectoderm. And the embryo is going to start to take on the architecture or symmetry of an adult. I mean, this is kind of what a sea urchin looks like. Mouth out here, anus up here. It's kind of a big spherical looking thing. That's more or less what's happening in gastrulation. You start to have the basic body plan of the adult. You've got all the germ layers you need, and you're good to go. So echinoderms, gastrulation is pretty simple. All right, same thing. This is just a micrograph of one of our embryos. You can see the archentron forming here, this little tube that's drawn in. The opening here would be the blastopore, and you still have blastocele out here. That's just a micrograph of a live one. All right, this is the frog. Now, the frog is still kind of a, spherical hollow ball like the echinoderm, but you had all this yolk in there, which kind of interferes. The cells can't just push right through it. Imagine it behaves sort of like a viscousy, syrupy stuff. It's hard to move around. So the cells just don't punch in. What they're showing you here, a little marker, the cells are beginning to kind of roll in around this surface. They form like a little crack and move in. That little crack is called the dorsal lip. That's an important marker on frog embryos. It'll also always be on the top because these things float with the yolk at the bottom because it's denser, the animal pole at the top. So the dorsal pole is the top one. And as the cells fold in, they form a little slit on the outside, which will eventually grow and wrap around until you have a completely circular opening, just like in the sea urchin. But the cells have to roll in around the yolk. They didn't just push in. And you end up, again, with the archentron here. And you got the 3 germ layers formed as the cells rolled over the dorsal lip. That signals them to turn into or develop into mesodermal cells. Now, the blastopore is right here, but it's only a potential opening. It's full of yolk, and they refer to that as a yolk plug. In fact, this stage in labs is called the yolk plug stage or gastrula. Either one will do. So a little harder for the cells to invaginate in for gastrulation in amphibian, but it's still more or less a lot like the echinoderm. Next, we'll look at the amniote lineage. We'll look at a chicken embryo, which will develop just like a human. It's completely different because remember, the embryo in a chicken or a mammal, the amniotes, isn't a ball like it was in the frog and the sea urchin. The embryo itself is just a sandwich. It's 2 flat layers of cells. It doesn't look like a ball. So while the stages are comparable, they don't look the same. The process is a little different. We'll look at that one last. All right, this is showing you kind of a 2.5D semi-cross section of a chicken embryo. It could be a human embryo as well. It's an amniote. We develop almost the same as a chicken. Again, because they have that massive amount of yolk. That's not going anywhere. So the embryo just develops as a little disc on the top of the yolk. Now, the way you can think of it, let me draw a line up here. What you have on this diagram, if I simplify it, you've got a layer on the top that is the presumptive ectoderm, and you got a layer on the bottom, which is a presumptive endoderm. It's what they're calling epiblast, and the hypoblast down here. But they will be the ectoderm and endoderm shortly. It's only 2 layers, and you're kind of like a sandwich. So what's happening, the cells, as these kind of arrows are trying to show you, the cells on the surface here, they're migrating this direction, kind of towards the midline from both sides. And when you look at this on the micrographs, as they move in the middle, they kind of pile up right along here and right along here. So on a slide, they'll look like 2 kind of slightly darker lines because the cells are thicker. They call this a primitive streak. This primitive streak is comparable to the blastopore in the echinoderms and the frogs. It's where the cells are going in, but there's no real hole there. So they're called a primitive streak. And as these cells push in, as these arrows indicate, some of them break off and move to this inner space, which would be on my diagram right here between the 2 layers. Now, as they break off, they're actually going to form the 3rd embryonic germ layer, which is a mesoderm, usually shaded in red. So all these cells were up on the surface. They've migrated in, and these little red shaded cells will form the 3rd embryonic germ layer, which is the mesoderm. So you'll end up with mesoderm here, ectoderm here, endoderm down here at the end of gastrulation. Now, the last thing to occur, well, not the last, but the next thing, this thing's got an architecture like a sandwich. Ectoderm on the top, mesoderm in the middle, endoderm on the bottom. That's not what you look like. You don't have ectoderm here, mesoderm in the middle, endoderm on the back or anything weird like that. This doesn't have the architecture of adult. So we'll look at the next slide. It'll kind of give you an idea. But what's going to happen, more or less, is this embryo is kind of going to roll up into a tube. So the edges will kind of roll down and seal at the bottom, and that's going to form a tube-shaped embryo, which is what we have. We're a tube. We got a hole here, a hole at the other end. Kind of a tube inside a cylinder. All right, so here's what you've got. There's your little three-layered embryo: ectoderm, mesoderm, and endoderm. Again, it looks like a sandwich. That's not what we look like. But what's going to happen if you look down here? See how it kind of rolls over, and it's going to fuse shut here. It's going to give you the architecture of a tube. That's our adult architecture. One of these openings at one end will be the mouth. The other one will be the anus. You have the dorsal aspect, the ventral aspect where it's sealed shut. So you have the basic symmetry of the adult body plan. That also happens in gastrulation. So you get the sort of adult body plan right after or at the end of gastrulation. All right, I think we got one more thing and I'll wrap it up. All right, this is showing you a frog embryo. So this is a frog down here. It could be a human, it could be a chicken. All of the chordates develop the same pattern. This thing is called neurulation. It's really the first organ system I'll show you develop, and probably the last one we'll look at. But what you'll notice at this point, after gastrulation, you got a cylindrical embryo, but all the cells still look the same. They haven't specialized yet. We know they're going to develop into different things, but they haven't expressed those differences yet that you can see. So the first thing you'll notice is up in the dorsal aspect, I'll mark it right here, some of the mesoderm just kind of condenses out and it forms a structure you can see. This is called the notochord. It's a kind of a hollow cartilaginous rod, acts as the main axial support for the embryo. In an adult vertebrate, that notochord is replaced by the vertebral column. So that's in the same spot, kind of has the same function. There's remnants of it in our intervertebral discs still, but most of it has been replaced by bone in the vertebrates. But that's the first thing you notice develop as a notochord. Then what happens is kind of cool. Over top of the notochord, this is the ectoderm. I'll kind of shade it in green. The ectoderm lying over that notochord signals it, and it starts to form what's called the nervous system. The ectodermal cells will thicken here, forming a thick area called the neural plate, still on the outside of the embryo. Then you'll notice in this next slide, the neural plate starts to sink in. It kind of forms this V-shaped bit here, and that's the neural groove. So it's kind of sunk down like this, and it'll keep sinking down, and eventually the edges, what they call neural folds, will fuse together. So if you look at the next slide, here it is. Those folds have fused together. It's sunk in. Whole thing's still covered with ectoderm. So that neural plate folded in, kind of rolled into a tube, kind of sunk in a bit, ectoderm covered it. That neural tube, that is your brain and spinal cord. Now, it starts off as a thin-walled, hollow thing, and it'll mostly fill in with cells and be mostly closed. But the hollow space is still present. The ventricles in your brain are remnants of that. The central canal that may be in the spinal cord is a remnant of the way this neural tube forms. And this is unique to chordates. You wouldn't see this in the echinoderms, but you would see it in the frogs or the chickens. And if there's a defect here, like spina bifida is a neural tube defect. If it doesn't close up all the way, you got a bit open, it causes some problems. But that's the last one we'll do, and I'll tack another on at the end. Again, this is just kind of a quick rehash of what we did in the lecture. I went in a little more detail, more examples, but hope this will give you a little bit to go on. Like I said, it's mostly for John because you missed. Take care.